around 2000 when the COVID-19 start showing up. Uh, uh, the number actually at that time in March 26, 2020 is almost 35 million. Of course, the number of this is not that much compared to the earlier outbreak of disease, but it's actually year by year that this start increasing. But in 2022, the late 2022 and 2023, the number actually start to be, it has been like a reduction because of the medication and vaccination too. So in 2020, this map shows the outbreak and the big problem of coronavirus. And as we can know, in USA, all the countries has a solid red color. It has the outbreak like USA, Brazil, Argentina, South Africa in Africa, Saudi Arabia, Russia, India, Iran. All this country actually is showing outbreak, and the number of cases and deaths is really high. And of course, in Europe, in Italy, and certain countries in Europe too. The situation actually from 2020 until 2023 until now, in the beginning, the number of cases and the number of deaths is really high. And as we can remember, the state of emergency actually in all over the world. But between 2021 and 2022, the medication and vaccination in certain countries start to be available. And based on that, the number of deaths and the cases start to be decline. And in 2023, the number really down because of the vaccination and medication all over the world, the first shot and the second shot. And in certain countries, there's the third shot actually of vaccination. And that's the case. The number is went down. In this study, actually, and as we know, hydrochloroquine and azithromycin is a basic two compound in every protocol for coronavirus all over the world, and millions of people took this two medication for recovery from coronavirus. And the mode of action of the hydroxychloroquine is create acidic environment to inhibit the replication of different virus. That's the mode of action of hydroxychloroquine. Ivermectin actually is another medication, and it's a famous one, and is written for all the protocols all over the world. It inhibits the nuclear import of viral and host proteins. And of course, the antibiotic azithromycin acts as a cytotropic lipophilic weak base, and that's the mode of action of the three compounds. In this study, we will focus on the hydroxychloroquine and azithromycin. So in 2020, between February and March, the number of the prescription for these two medications, hydroxychloroquine and azithromycin, is got increased by more than 1,000%. So the general hypothesis actually in this study is, are azithromycin and hydroxychloroquine predict any negative impacts on the health of the study's animal model or in the environment in general? That's our general hypothesis we are testing in our study. The aims of this study is actually two aims. The first one is predict the impact of the indiscriminate use of azithromycin and hydroxychloroquine in this pandemic context. The second one, we try to assess their toxicological risk when isolated or in combination, the combination between the azithromycin and hydroxychloroquine, and we're using the zebrafish Danu as a model system. But the question is why we are using zebrafish. Uh, Actually, zebrafish is a good model actually for the toxicologicalist scientist, and for many reasons. Actually, it has many advantages. The first one, external and rapid development, short reproductive cycle, high number of spread. Genes have a true orthology in the zebrafish genome. Zebrafish genome is fully sequenced, and all this advantage actually we used why we are using zebrafish in our study. So as I mentioned before, we use two compounds, is very well-known compounds and protocols for recovery from the coronavirus. And in this study, we use azithromycin concentration 500 milligram and hydroxychloroquine 400 milligram. And we use the concentration of 12.5 microgram per liter from each medication. The model system and the experimental design, we use 96 healthy adults divided by 4 groups. Each group have like 24 fish, 3 replicate, and 6 animals. And it has both sex, male and female. The 4 groups actually, the first one is control. The second one is for azithromycin. The third one for hydroxychloroquine. And the fourth one is the mixture between azithromycin and hydroxychloroquine. The results has been taken after 72 hours. And we assess the many nutrition status actually. For example, the total protein, triglyceridine, and total soluble carbohydrates level. All this actually nutrition status actually we measure after the 72 hours exposure for the each one alone, the azithromycin and hydroxychloroquine, and the mixture between both. Oxidative stress biomarker, it has been actually tested in this study actually, and we evaluated based on the first one, indirect nitric oxidase, the second one, thiobarbituric acid reactive substrates, and the third one, the production of reactive oxygen species, ROS. The silico-chemogenomic based, we're using the ChemDes system analysis. And at this, actually, we use gene expression actually to get the effects of many genes in our study and the effects of azithromycin and hydroxychloroquine on it. The interaction network analysis, we used a high developed methods actually. It's called network analysis. It shows the interaction between the medication we are tested in this study and different enzymes, especially the B450 enzymes, and it shows the reaction by color. For example, the binding given blue color, activation given green color, inhibition given red color, catalysis given magenta color, reaction given black color. And we shown in the figure as the network interaction based on each color actually. The molecule docking actually we using to predict the binding size and affinity of the bonds among the azithromycin and hydroxychloroquine for the protein structure, especially for the enzymes we are using here in this study. Acetylcholinesterase enzymes, the superoxidase dismutase enzymes, and many others, the catalase enzymes. And we're using the chemo informative screen for show the results of the molecular docking. Also, we test the neurotoxicity actually on the nerve system, especially for the spots in the head and the tail. As we can see actually in the left side, the diagram for the head and tails and showing the spots when we are testing our experiments, and the natural in the right side we seen actually the natural spots where it's figure out in the head and in the tails. And we use a statistical analysis. We're using one-way ANOVA and TICS post hoc analysis. All the graphs have been designed by using GraphPad Prism software version 8. And the correlation analysis, we're using Pearson test and Spearman test. As a result, and as we can see in the figure now, we can see the depression actually and declines that happen after testing the azithromycin and hydroxychloroquine. And as we can see, is not significant effect on the total carbohydrates in the figure A, but in B and C, there is a significant reduction actually in the total protein levels and triglyceride level in figure C. In this figure, actually, we showing the effects of azithromycin and hydroxychloroquine and the mix between each other on the oxidative reaction, especially on reactive oxygen species and superoxidase dismutase. And as we can see, the effects of the medication alone and in the mix, there is increasing actually on the oxidative interaction, and especially when we are testing the ROS, the reactive oxygen species is going really high, and nitric acid level and hydroxygen peroxidase and the cyobarbitic acid reactive, all these parameters gotten increased significantly compared to the control side. In this figure, we are test the binding energy between each compound and the enzymes for catalase and for superoxide dismutase. And we can see actually for figure A and B the amount of the energy that release after the interaction between the binding happen. Here in this graphs, we showing the networking interaction analysis. And as we can see, the colors, it's showing the network for the binding, for the activation, for inhibition, for the catalysis. And for example, the blue color, it's showing the binding between the compounds. And for example, in this study, the B450 enzymes family, and it's showing the green color, it's showing the inhibition between the enzyme and the compounds, and the green color showing the activation between the compounds and the enzymes family. Here we're showing the gene expression for the many genes, and we divided the genes here for three categories: biological process genes, molecular mechanisms genes, cellular components genes. So it's shown here the gene expression for the effects of azithromycin, and here for hydroxychloroquine alone, and here for azithromycin and hydroxychloroquine in mixture. So we divided the genes in three important categories: the biological process, the molecular mechanisms, and cellular components, and showing the gene expression for each gene. Here also we use the effects of the compounds alone and in the mixture on the nervous system, and the effects of acetylcholinesterase enzymes, the activity of acetylcholinesterase enzymes activity, and is showing actually the hydroxychloroquine in the figure A. There is a significant increase in the activity, whereas there is non significant effects between the control and the azithromycin, but both in the mixture between azithromycin and hydroxychloroquine, there is actually significant increasing on the acetylcholinesterase activity. And in the figure B, it shows the energy between that released between each compound and the enzymes. In this figure, we show the effects on each compound alone and the mix on the neuromax and the nervous system, A, the tail, and B for the head. And it's showing actually there is a significant inhibition, especially for the head, the neuromass that occur in the head more than in the tail, which actually figure out specific effects on the nervous system in the zebra fish. The summary of these results is actually for 5 points. We observed that 72 hours of exposure to azithromycin and hydroxychloroquine alone and in mixture induce the reduction of total protein level combined by level of thiopyruvic acid reactive substrates, hydroxygen peroxidase, reactive oxygen species, and nitric acid. Suggestion actually that a redox imbalance and possible oxidative stress. Number 2, the molecular docking analysis further supporting this data by demonstrating strong affinity of azithromycin and hydroxychloroquine with their potential antioxidant target, catalase and superoxide dismutase. In the protein-protein interaction network analysis, azithromycin shown a putative interactive with different cytochrome B450 molecules, while hydroxychloroquine demonstrated interactive with caspase 3. The function enrichment analysis also demonstrate diverse biological process and molecule mechanism related to the maintenance of redox uh, homeostasis. We also demonstrate an increase in the acetylcholinesterase activity, followed by a reduction in the neuromass of the head when zebrafish were exposed to the mixture azithromycin and hydroxychloroquine. And the data suggests that neurotoxic effect of the compounds occurred uh, in this study. The conclusion of this study actually shows that short exposure, even after 72 hours, uh, for the azithromycin, hydroxychloroquine, and the mixture, induce physiological alternation in the adult zebrafish, and the effects can compromise the health of these animals. And our suggestion actually is that increase of azithromycin and hydroxychloroquine due to COVID pandemic can negatively impact freshwater ecosystems and can has bad uh, effects and impacts actually on the human being. This study published in a journal and it published in 2021. And the authors actually, the scientists on this paper from USA, Dr. Christoph Vogel, and different scientists from Brazil, especially Professor Guilherme Malafia. Uh, Finally, I have to thank all the authors and all the scientists work in this study. And as we can mention before, uh, three countries working on this study: Egypt, USA, and Brazil, mostly from Brazil, because at that time, the number of cases and the number of deaths is outbreak. Is millions of people got this and got infected by the coronavirus. Uh, Thank you.
Thank you, Dr. Mohammed, for the nice presentation. And now we open discussion. So if anyone has a question, you can just raise your hand or write your question in the chat and to answer it. Any questions? Have something? Okay, Dr. Muhammad, we have a question from Yusuf.
Uh-huh.
I will allow mic. So unmute your mic, Yusuf, and speak.
Hello.
Hi.
Can you hear me?
Yes, the sound is like a far sound. So could you please increase the volume?
I guess.
Okay, yeah, go ahead.
Can you hear me now?
Yes.
Can we understand from this that the all drug or treatment used in used to treat coronavirus are harmful for people health?
I can't tell you exactly is that true or not until we have to test all the compounds you mentioned, like ivermectin. We didn't test in this study. But it sounds like, yeah, but we need to do experiments to be sure that these compounds has a bad effect of our environment. And in this study, we focus on the zebrafish, and it's showing actually really negative impact on this kind of fish. But we have to examine all compounds in the same way, maybe in different model testing, like fish, or maybe in rats, or mice, or different mammalians. We can test it. So it could be, but we have to test it to give you the 100% answer for your question.
This means that there are other compounds that does not cause harm for people, or this compound you need to examine on other animals.
We did in this study the examine our zebrafish, and as I can show, and we published our work, it's really negative effects. It could be the other compounds, it could be has the same like negative impacts or negative effects, but we didn't do yet. We need some other authors and scientists to test and examine the effects of this compound, not only in zebrafish. You have to use another models to be sure, this compounds can be effects in a bad way.
Okay, thank you.
Thank you, Yusuf. Do we have another question or?
No. Thank you.
Okay. Any question from other participants? Maybe can see in the chat. Okay, Dr. Mohammed, I have a question. So, how many genes you tested for the expression analysis used?
So it's almost like 25 genes.
Yeah. And I didn't just get is upregulations or downregulations due to treatments. The treatments affected on the genes, upregulation or downregulations.
Some of them is down, some of them is upregulations, and as we can see in the gene expression, some of them, there is downregulations, others is upregulations.
Okay. So, do you think that these genes can be also tested further in other animals, or just specific for zebrafish?
No, we can use it in other animals too. Remember that we're sharing with the zebrafish by 70% of the genes between human being and zebrafish. That's why we are focused on zebrafish. But we can use in other models actually, and we test the same genes, and we can see if there is upregulation or downregulation gene expression based on the compounds we are testing in this study.
Okay, thank you. We have Yusuf. We'd like to ask again question. So unmute your mic, please.
Hi, hello, it's me.
Yeah.
This compound have a mutant effect on cellular level.
Yeah. As I mentioned before, in the our study, we focus on the oxidative activities actually, and on the molecular mechanism too. It has like negative effects too, and the cellular components, as I shown actually in the figures that show the genes. It has some of them like got increased in the level of the gene expression, and others actually getting down in the gene expression. So there is effects on the cellular component.
No. I talk about mutation.
Mutation?
Mutation.
You mean mutation?
Mutation. If this compound effect on has a mutant effect.
We don't test the mutation yet. We only focus on the gene expression, and we didn't go further actually. Maybe in the next work or next study, we focus on if this compounds make like a mutation. And actually, this is a good point. Thank you.
Okay, thank you.
Yeah, it's a really good question. So
Yeah,
to see the effect of this
happens the mutation or not.
Yeah, on the DNA of the zebrafish or not.
Yeah.
Okay. So no question, no further question. So by the way, I would like to thank Prof. Mohammed for the really enrichment seminar, and I know that we got very useful information for the first time for me as well. And I would like to thank the participants for showing up today. And thank you, Dr. Mohammed. Please, if you'd like to get a certificate of attendance, please fill the form of registration link, and we will issue the certificate after the end of seminars. Maybe in August, we will send you an email regarding to this issue. And you don't need to attend further seminars. Just attending one seminar is enough to get the certificate of attendance. But maybe later in August, we will inform you about this, how to get the certificate of attendance. Thank you for all, and see you next week in Wednesday. We have interesting lecture in plant genetics, a scientist from Ethiopia. So see you next Wednesday. Thanks for all.
Thank you very much, Dr. Ahmed. Thank you.